So this is always exiting. And these are our motor neurons that are going to be able to bring out our motor commands, voluntary or autonomic, to the effectors. Alright, what's the relationship of a nerve to a neuron, or a neuron to a nerve? Neurons are the tiny little signaling cells. And so the relationship of a neuron to a nerve is much like the relationship of a muscle fiber was to a muscle. So we said that we had a big muscle that was surrounded in epimysium. Well, we've got a big nerve that's surrounded in epineurium. Epineurium is analogous to epimysium. So what kind of tissue is it? Dense, irregular. So we'll have epineurium surrounding whole nerves. Whole nerves contain many fascicles that are surrounded in perineurium. What do you think perineurium is made of? It's also dense, irregular. And then each of our muscle fascicles will have, each of our nerve fascicles will have many neurons. We can see the axon of this neuron coming out here. And each of our axons and neurons are surrounded by endoneurium that is going to electrically insulate every neuron from its neighbor neuron. So what's it made of? Areolar connective tissue. And that's going to surround our individual neurons and keep them insulated from each other. So in our nerves then, we'll have many, many neurons. And in spinal nerves, all of our spinal nerves are mixed. They've got neurons of both sensory and motor neurons. We can see, if we look here, all of our sensory neurons would be coming in this posterior region. We can tell because of this posterior root ganglion. And then all of our motor neurons are going to be coming out in the anterior region. So all of our spinal nerves are mixed nerves. They've got both motor and sensory fibers in them, or motor and sensory neurons in them. In the cranial nerves are the nerves that are coming out of your brain, and they can be sensory, motor, or mixed. So we'll look at that with the next chapter. We'll look at those in all more detail. So nerves are then these big structures that contain many neurons. The neurons are gathered in fascicles, and we've got these same connective tissue regions that are holding them together, similar to the muscle. So if we look at when we see a spinal nerve exit the spinal cord, you'll have this posterior region that has this ganglion. A ganglion is a group of cell bodies. In the spinal cord, we call it a posterior root ganglion because you see it in the posterior root of the nerve. And then the nerve has both sensory and motor neurons in it. All right, any questions so far?
Ganglions, would you find those anywhere or just with the spinal?
So you can find ganglion coming up around and out of your cranial nerves. So we'll look at those, we can see those. In the spinal cord, they're really easy to recognize. They're really right there close to the spinal cord. So, and then we'll see, we've got sympathetic chain ganglion that are going to be part of the autonomic nervous system. So that will cover all on its own after we get through the brain and cranial nerves. So for the next test, for the last test, it'll all be about spinal conduction pathways, and we'll look at the autonomic we'll specifically look at the branches of the autonomic nervous system, which are going to be really important for 202. And then we'll finish up talking about senses. So yes, you can find them other places, but in general they're located right next to the spinal cord. Any other questions? Okay, so let's look at our glial cells. Glial cells are the cells that support our neurons. The first type that I want to talk about are these guys called astrocytes. And they're really probably, I don't know if I want to say most important, they're definitely the most abundant, and they're very important. They serve many functions for neurons. So astrocytes are our most abundant glial cell, and they're going to do several important things. So functions of astrocytes. First, they're going to help form the blood-brain barrier. And the blood-brain barrier is this barrier that prevents the passive passage of materials from the blood into the brain extracellular fluid. So that's its function, is going to prevent passage of material from the blood to the extracellular fluid. So that's their first important function. And we have these things called perivascular feet. Peri always means around, vascular being vasculature, and these perivascular feet wrap around the blood vessels and create this tight space so that anything that's coming into, I shouldn't say anything, most things coming into the blood extracellular fluid have to get through the astrocytes first. So the astrocytes are preventing just the passive passage of stuff like some virus or bacteria that's floating through your blood. Can't just passively get into your brain because we've got this blood-brain barrier. So that's their first important job. Then as part of that, they're going to help regulate the composition of the extracellular fluid. So they regulate the composition of the ECF by taking in like ions from the blood and getting it out here to the extracellular fluid if need be. If we've got like high levels of potassium or something out here, they'll take it up. Potassium is really, really important to keep in low concentration extracellularly so that we have this difference, that sets up our resting membrane potential. So if you have an increase in potassium in your brain extracellular fluid, then the astrocytes can take it up to make sure you don't start messing with your resting membrane potentials. So that's going to be another important function. They're also going to help to form the framework for your neurons, and they guide normal neural development. You can look at these brain models and they all look the same. And if you cut open cadavers and look at their brains, unless you're Einstein or something and have a big physics region, most brains look the same from brain to brain to brain. So that happens because our astrocytes help to form the framework and help with the growth and development. So we'll say they form the framework for our neurons, and they guide them during development. So they guide normal development. So astrocytes are pretty darn important. And then the last thing that they do, we said that you have many neurons die off at these clippings that you're not using. Well, astrocytes fill in the space of dead and dying neurons. So astrocytes are very, very, very important for the brain. The next cells that we should talk about are these ependymal cells. I don't like that they have the ependymal cells right here next to this blood-brain barrier that we're forming with the astrocyte, because ependymal cells are found in the choroid plexus, which is this specialized area that's free of blood-brain barrier. And the reason it's free of blood-brain barrier is because we've got to be able to, these cells have got to be able to get things out of the blood to make our cerebrospinal fluid, it's called. And the cerebrospinal fluid circulates through these empty spaces in the brain called ventricles, and it circulates around the brain and then down in through your spinal cord. So if they couldn't have access to the blood, then they wouldn't be able to make cerebrospinal fluid. So our ependymal cells then are these epithelial-like cells that make cerebrospinal fluid. And we'll talk about the functions of cerebrospinal fluid in the next chapter, I believe. So they make cerebrospinal fluid, and they're found in this place called the choroid plexus, lining all the ventricles of our brain. Other glial cells, the next glial cells that we'll talk about are these microglial cells. Microglial cells are phagocytic. So these are the only local immune cells that the brain has. So they will gobble up debris, anything foreign objects running around in there. So, I mean, not that you should have a lot of foreign objects running around in your brain, but if you've got dying neurons and things, then all of that debris can be eaten up by the microglial cells. So these are really the only local immunity that the brain has. And then the last type of cell, glial cell, that we have is the oligodendrocyte. These are all the glial cells that we find in the brain. And the oligodendrocytes are these cells that have this important job of creating what's called a myelin sheath. The myelin sheath is like this insulating material that forms around the neurons, and it helps to speed up the conduction of nerve signals. So just like copper wires have to be insulated so they can speed up transmission, neurons have to be insulated so they can speed up transmission, and the oligodendrocytes are the cells that do it in the central nervous system. So they're going to be our myelinating cells in the brain. In the peripheral nervous system, we only have two kinds of glial cells. These cells that are found around the cell bodies of our sensory nerves, and they're going to be called satellite cells. And satellite cells they're not doing nearly all the cool stuff that astrocytes do, but they're kind of helping to regulate the different materials that are entering and exiting the cell body, and they can kind of help regulate what's going on in the extracellular fluid. So they regulate materials coming into and out of the cell bodies in our peripheral neurons. So they're forming this barrier around the soma so that nothing can just get from that extracellular fluid in without passing through them. And then the other type of glial cell in the peripheral nervous system is the cell that your book calls a neurolemmacyte. These are also called Schwann cells. Most books, I think, still call them Schwann cells. And Schwann cells are the myelinators in the peripheral nervous system. And myelination in the peripheral nervous system is really important because it's speeding up the conduction of our action potential. You've got some nerves that are really, really long. Neurons running in your sciatic nerve. It's the biggest nerve in your body and runs from your lower spinal cord, like exits near those branches that are exiting near the sacrum are going to fuse into this sciatic nerve that runs all the way to the bottom of your foot. And you can wiggle your toe as quick as you think about it. And so in order to do that, to get that message down that quickly, we have to have these neurolemmacytes providing these myelin sheaths because it really speeds up the rate of conduction. So the lady who teaches before me drew this picture up earlier, and I just left it because it's so beautiful, of how these myelin sheaths work. And what it does is that if you look at a myelin sheath, then all of the spaces, or if you look at a myelinated axon, all of the spaces in between the myelin sheaths are what are called nodes, or nodes of Ranvier. And when the action potential is coming down the neuron, what happens is that this space is insulated in here, so you don't have this loss of ions, and basically all of those sodium ions that rush in as part of our action potential can be maintained in here. They're not getting kicked out really quickly by that sodium-potassium pump because we're insulating it, we're protecting it. So that the action potential, if you measure it, it looks like it jumps from node to node to node to node to node. We call that saltatory conduction, and it speeds up the rate of nerve transmission. So it's so important because it does what's called saltatory conduction, and it speeds up the rate of nerve transmission.
Now, is saltatory conduction speeding up action potential when it's speeding up the rate?
Basically,
Yeah.
it's because the action potential is jumping from node to node to node to node. It doesn't have to replicate in all these regions between here. So it's making it go faster. So the action potential is going to be the same magnitude all the time, but our myelinated neurons are going to be able to send the action potential much, much faster. People who have MS what MS is, it's a problem with myelination. And they'll start to lose their ability to control their motor commands as quickly, and it's because they're losing this myelin. So how myelination occurs is you'll have a neurolemmacyte will begin to wrap around an axon, and it continues to wrap around, so it's making more and more neuroplasm and more plasma membrane, and it wraps around and around and around until we get this nice big thick multi-layered insulator around the axon. And that's really going to help speed up the rate of transmission. In the peripheral nervous system, neurolemmacytes or Schwann cells are the cells that are making the myelin sheath. In the central nervous system, it's oligodendrocytes. And they're reaching out extensions that can branch around many neurons, several neurons in different places to make many myelin sheaths. Okay, then the next thing we need to talk about is what your book calls the ultrastructure of neurons. So what do they look like at the microscopic level? We met all these things a little bit in the muscular system. So first, all of our cells are going to have the sodium-potassium pump. It's going to be a pump because it's moving things against their concentration gradient, so it has to use ATP to do so. So in normal circumstances, sodium is much higher outside the cell. Potassium is much higher inside the cell. So the sodium-potassium pump is going to use ATP to pump these things against their concentration gradient. So it'll bind ATP and 3 sodium ions will enter, and then it'll split the ATP, pump those 3 sodium ions out, and bring 2 potassium ions in. So the sodium-potassium pump sends in 3 sodium ions and sends out, wait, I just said that wrong. It sends out 3 sodium ions and is bringing in 2 potassium ions. So it's going to pump these things against their concentration gradient. So this will be how we get, after we have this rush of sodium into the cell and this rush of potassium out of the cell, how we get them moved back against their concentration gradient. Then all of our neurons also have these leak channels. Leak channels are passive. They do not require ATP. They're just stuck in the cell membrane and allow ions to flow down their concentration gradient. So we've got, this is showing us sodium leaking into the cell from area of high concentration gradient to low concentration. And then potassium leak channels would be working the other way. Potassium would be leaking from high inside to low outside. And it's the differing number of these leak channels that is going to set the specific membrane potential, different resting membrane potential for different cells. So in our muscle cells, the resting membrane potential was negative 90 millivolts. There were a lot more potassium leak channels in our muscle cells than there are in neurons. The resting membrane potential of our neurons is negative 70 millivolts. Now, why is it negative? Well, the cytosol has lots of proteins that have negative charges, and there are more potassium leak channels than there are sodium leak channels. So potassium is a positive ion. So if it's leaking out more than sodium's leaking in, that's going to help drag to this negative resting membrane potential. Other types of channels that are important are these chemically gated channels. The chemical in this case is a neurotransmitter. So our chemically gated channels exist in 2 states. They have a closed state when they have not bound their neurotransmitter, and an open state when they have bound their neurotransmitter. So this is showing us a chemically gated potassium channel. So when the neurotransmitter binds, potassium will then go down its concentration gradient and leave the cell. Other types of channels that are going to be important are the voltage-gated channels. We talked about these before, but we didn't talk about the specifics. So now we need to talk about the specifics. So voltage-gated channels exist in 3 different states. They have a closed resting state where they're just waiting to be stimulated. And the stimulus that's going to open them is a specific voltage, specifically negative 55 millivolts. is the threshold that opens them. So then that's the key that opens the door. So when they hit -55, they open and enter this what's called activation state. When they're open now, their ion can flow down its concentration gradient. So sodium will dump into the cell, potassium will dump out of the cell. Then they have a third state, this closed inactivation state. And I think of the inactivation state kind of like, okay, if we've got a closed door, right now these doors are closed, you could open them and you could rush out from high concentration to low concentration. Okay, but now if I run out and I stick on like a bunch of 2x4s and I bang them in and nail them in so you cannot get out, then you can try and open the door, but you're not going anywhere. That's what the inactivation state is like. So after the action potential is coming, when we hit that positive 30 millivolts for our sodium channels, those channels close and they enter their inactivation state. And during the inactivation state phase, there's no amount of stimulation that we can do that will open these gates. And then a little bit of time will pass and we'll come back to the resting state. And what this does is ensures the one-way movement of the action potential down the length of the axon so that it doesn't go backwards. So if we look again, our neurons have all of these, and they're going to be helping with the different parts of what's important for the neuron. So these three images right here are what's setting the resting membrane potential. So your differing number of leak channels, both sodium and potassium leak channels, and your sodium-potassium pump is setting the resting membrane potential. In neurons, it's -70 millivolts. These chemically gated channels are responding to neurotransmitter. So these are what allows our neurons to be excitable. These voltage-gated channels are found all down the length of the axon until we get to the terminal, and these are what's allowing the cell to be conductive. So if we look at a whole neuron, down the entire length of the neuron, we've got sodium-potassium pumps, sodium leak channels letting sodium leak into the cell, and potassium channels letting potassium leak out of the cell. So that's found down the entire length of the neuron. Those other channels are only going to be found at specific parts. So it's going to give us specific different regions of the neuron that are going to be able to do different things. So up here, at the cell body and in our dendrites, we've got chemically gated channels. So these are all going to be our neurotransmitter gated channels. The chemical that's binding is a neurotransmitter coming from some other neuron. We've got the chemically gated cation channel that's going to allow for the movement of cations with a net influx of sodium in. We've got our chemically gated potassium channel that's going to let potassium out. The chemically gated cation channels are going to let sodium in so that we'll get closer to the threshold. The chemically gated potassium channel is going to let potassium out so we'll move further away from threshold. And then we've got a third type of chemically gated channel, the chloride ion channel. And chloride ion is higher, it's a negatively charged ion, and it's in higher concentration outside the cell. So if a neurotransmitter binds that, then chloride ion is going to enter the cell and also move it away from threshold. So we'll talk more about all these things in a little bit, but just be aware that the receptive segment up here, where we've got the dendrites in the cell body, only has these channels. And it's important that these are the only channels because this is the only spot we want responding to neurotransmitters. And we don't want voltage-gated channel opening here, because that's going to be how we get our action potential to move. Okay, this next region we have is what's called the initial segment, or the axon hillock. And the axon hillock is like this enlarged region coming off of the soma. And the channels that we have there are voltage-gated sodium channels and voltage-gated potassium channels. So this will be the first place that we can start our action potential. And once it starts, it's going to go. The action potential is an all or nothing event. So as soon as we're going to add up all of these things that are happening here in the soma, and if we hit threshold, we're going to open those voltage-gated channels and the action potential will proceed. The next region of our neuron is called the conductive segment, and it's conducting the action potential down the axon, and it also has voltage-gated sodium and potassium channels. The conductive segment actually goes all through the telodendria until we get to the synaptic knobs. This picture's calling them synaptic bulbs. They're also called axon boutons because they look like buttons. So the synaptic knob is going to be that transmissive segment where the only channels that we've got present are the voltage-gated calcium channels, and then we also have to have a calcium pump because after all that calcium dumps in, we got to be able to get it back out so that calcium's in higher concentration outside the cell. Yep.
You say that the sodium potassium pump is everywhere.
It's everywhere, yep. As are the sodium lead channels and potassium lead channels. So they're found along the whole length of the neuron. Alright, so a potential is a difference in charge from one side of the membrane to the next. So that's going to just be what the general definition of a potential is going to be, a difference in charge. From one side of the membrane to the next. So if you stick an electrode in this neuron and it's at rest, we'll see that the resting membrane potential of the neuron is negative 70 millivolts. And that is set up by the sodium-potassium pump and the sodium and potassium leak channels. Again, there are more potassium leak channels than there are sodium leak channels. So you have this positive charge leaving. We've got all these negatively charged proteins in here that are keeping it negative in here. You've got lots of potassium leaving. So our resting membrane potential is negative 70 millivolts. If we look, we can see that we've got different concentrations of these different ions. Sodium is higher outside the cell. Chloride ion is also higher outside the cell. Another one that's higher outside the cell but not important for most of the neuron is calcium. It is very important when we get to the synaptic knob. Calcium ion is also higher outside the cell, and potassium ion is higher inside the cell. So movement of these ions is what's going to be allow the neurons to conduct this electrical pulse that we can read and we can measure all down the length of the axon. So our resting membrane potential is negative 70 millivolts. If we have a movement away from rest as sodium flows in, that's depolarization. So a movement away from rest in the positive direction is depolarization. So we'll say move away from rest in the positive direction. And depolarization will be due to sodium coming into the cell. So even if you don't hit threshold, as long as you're moving away from rest in the positive direction, that's depolarizing. Now what about if we move away from rest in the negative direction, either by opening a gated potassium channel and letting potassium leave, or by opening a chloride ion channel and letting chloride ion enter? That's what we call hyperpolarization. And in hyperpolarization, you're moving away from rest in the negative direction. So again, our resting membrane potential is negative 70. If we move away from negative 70 in the positive direction, then we call that depolarizing. If we move away from rest in the negative direction, that's hyperpolarizing. And we can hyperpolarize due to potassium out or chloride ion in. Any questions so far? Alright, so we're going to break our neuron down into these different segments of the cell depending on what each segment is doing. So this receptive segment is going to be where we've got those neurotransmitter gated receptors, And they're going to open up to either allow a net influx of sodium or to allow chloride ion in or to allow potassium ion out. What they're doing is producing what are called graded potentials. Graded because they decay over time. So this receptive segment has our chemically gated channels that create graded potentials. So we'll have a whole bunch of other neurons synapsing onto this guy, and it's got to take all of those potentials and decide what to do with them. So the region of the neuron that decides what to do with them is this initial segment, and it's going to sum up all of that stuff and decide whether or not we initiate an action potential. If we hit threshold, we will initiate an action potential, and it happens all or none. If it's going to happen, it's going to happen. It's going to conduct down the whole conductive segment and cause the release of neurotransmitter. If we don't add up, then we won't have an action potential. So the initial segment is where we're going to sum those graded potentials. And then if we have an action potential, if we initiate an action potential, it will be conducted down the conductive segment, which is most of the axon and those telodendria that are leading to the synaptic knobs, and it's propagating the action potential. The action potential is self-propagating. Once it starts, it doesn't need anything else. And then once the action potential gets down to the transmissive segment, that's where our synaptic knobs are. The transmissive segment, it's going to ultimately cause the release of neurotransmitter into the synaptic cleft.